Great. Thank you so much. Can you guys hear me? Thank you so much for the nice introduction. And it's really great pleasure to be here to talk about our research. Today what I'm going to do is to talk about a few different statistical methods our group recently developed for analyzing spatial transcriptomics. So spatial transcriptomics is a very new set of new technologies that just come about 3, 4, 5 years ago. And this is technology that's currently revolutionized the entire field of biology. So as many of you probably have heard of bulk RNA sequencing. So bulk RNA sequencing was a technology developed by about 15 years ago. So that's a technology where you can get a tissue, you can homogenize a tissue, extract the mRNA, and measure the gene expression level for 10,000 genes. Now, the bulk RNA sequencing is a powerful technology. The first time allow you to digitalize and measure accurately the gene expression level across the entire transcriptome. But it has a very disadvantage. Very unique disadvantage is that you are getting a tissue, you are measuring a tissue, and the tissue consists of thousands of cells that are potentially coming from different cell types. So you are measuring the average expression level of the tissue, which is not of ideal, not of a high resolution. So if you think about analogous to a painting, pointless painting, you're basically putting all the different colors mixed together, putting them together into a picture. So that's your bulk RNA sequencing. Not very high resolution, but you did measure something. Now, 10 years ago, there's a new technology, set of technology developed. It's called single-cell RNA sequencing. So single-cell RNA sequencing is able to extract every single cell from that tissue and measure gene expression level. So that gives you extremely high resolution, measuring across tens of thousands genes on each and every single cell. In terms of an analogy to a painting, this time now you are able to sort out every single pixels out, sell them separately, allows you to characterize better the tissue information. However, one disadvantage of single cell is that it does not have the spatial resolution. You are not able to measure which single cell came from which location of that tissue. So the spatial transcriptomics are a set of new technologies that just recently developed, allows you to finally analyze measure the gene expression level across thousands or even nowadays millions of spatial locations on the tissue to better characterize the architecture of the tissue. So in terms of painting, now you are able to put all the correct color into the right spatial spots to form a nice picture. So there are many different spatial transcriptomics technologies developed. There are two different large categories. One categories are based on sequencing. The other categories are based on SNFISH or single molecular first in situ hybridization. So the sequencing-based technologies include early on the spatial transcriptomics, which becomes the commonly known 10x genomics within platform. There's also other commercial platforms such as a GeoMix and other more recent high resolution technologies such as SLAD-seq, high definition spatial transcriptomics, and also seq-scope, which was developed at the University of Michigan. So those are the sequencing-based techniques. The other SNFISH-based techniques include MERFISH, which was also recently commercialized, seqFISH, seqFISH Plus, and CosMx. CosMx is another commercial platform. So let's look at two examples, see what data comes from, so that we have a better idea how to better model the data. So let's look at this 10x vision technology. So in the vision technology, you have this commercial prepared slides. On the slides, you usually have 4 different squares. Those are the capture area. So if you are interested in the tissue, so let's say you are interested in measuring gene expression on this half section of this mouse brain. So you can overlay the mouse brain on one of those squares. And on the squares are very unique because it's plated with a lot of different spots. So if you zoom in, you will actually see about 5,000 to 6,000 spots on this the squared captured area. And each spot consists of multiple probes. Those probes are very unique, contains many important features that allows you to measure gene expression level. So specifically, those probes have this poly T tail, which allows you to bind the released mRNA so that you can measure gene expression. So little UMI count, the unique molecular identifier, allows you to cleanly measure the gene expression without any PCR artifacts. And then finally, the most important part is the spatial barcode, which is unique for spatial transcriptomics. So the spatial barcode, once you sequence it, it allows you to map back the reads back to the spatial location so that you effectively extract the X and Y coordinates of each measured spot. So therefore, at the end of the day, you are going to get two matrices. One matrix is N by P matrix, which is usual in terms of genomics measurements. N is the number of spatial locations you measure, P is the number of genes you measure. But in addition to this N by P, which is a count matrix here since it's a sequence-based, you also get another matrix, that's an N by 2 matrix. That's the X and Y coordinates for each of every N locations. So that's unique to spatial transcriptomics. So those are the two metrics you can get for 10x genomics vision. These are our basically data input. Certainly for the SM fish-based technology, you are essentially getting a very similar input data. So like in the SIG-fish plus technology, you are essentially measuring gene expression through a sequence of hybridization. So you hybridize, wash out, hybridize again, and each hybridization you are throwing a different color barcode. So therefore, using a different color combinations, you are able to measure each and every single gene. So with the SIG-fish plus, you are able to measure 10,000 of genes, and at a very high resolution, you can capture the molecular level at the single cell resolution on the spatial plate. So at the end of the day, with the SIG-fish plus technology, you're also going to get two matrices. One is the N by P, where N is the number of single cells now, P is the number of genes. It's also a count matrix because you're measuring those different color dots within a single cell boundary. And besides that, you also get the N by 2 matrix, the location matrix, where you have the X and Y measurements for each of the N different cells. So regardless of which technology you have, you are always going to have these two output or input matrices. One is the N by P count matrix of gene expression, the other is N by 2 location matrix. And certainly, sequencing-based and SM-fish-based, they all have their own unique advantages or disadvantages. So with this two input matrix, we can start to answer a lot of important scientific questions using statistical modeling. And today I'm going to talk about the two major applications. One application is for so-called SE analysis or spatial expression analysis. The goal is to identify spatially expressed gene. The other, if we have time, we're going to talk about doing cell type deconvolution. So SE analysis is essentially analysis trying to detect SE genes that display spatial expression pattern. So SE genes are also commonly referred to as SVGs or spatially variable genes. This is very similar to doing a DE analysis in bulk or single cell RNA sequencing analysis where you try to identify differentially expressed genes. So here shows one example of SE gene. So here, this is the olfactory bulb tissue slides. So you have, it's a mouse, so you have two different olfactory bulb. It's symmetric, so rounded, circled olfactory bulb tissue section. And each dot is a measured spot. And we're only visualizing one gene here, and the color visualize the intensity or expression level of that gene. So specifically, the pink means that the gene is heavily expressed, and the green means it's lowly expressed. So for this particular gene, you can see that it's heavily expressed in the middle of these two olfactory bulb. And our goal is to identify those genes that show this type of unique spatial pattern. This is in contrast to any random expression pattern you might expect from housekeeping genes that you would expect to see a sort of random homogeneous spatial expression pattern. So there are, when we first start to look at this type of data, there were already two methods developed. One is called Transcriptomics, the other called spatial DE for SE analysis. But there are important limitations. Specifically, they usually model normalized data instead of count data, and so they are going to lose accuracy and power. Also, the p-value computation is either inaccurate in terms of the spatial DE because it's using the asymptotic form, which does not kick in that accurate that well over there, and also it's very slow in terms of Transcriptomics method. So therefore, motivated by this, we decided to develop a method called Spark, or spatial pattern recognition by kernels. So this is based on generalized linear spatial models. So we're going to examine one gene at a time, try to detect whether the gene shows any spatial pattern or not. So to get there, we will use some notations. So we will use S sub I to denote the spatial coordinates for each of I's location. So you have n different locations here. So those are basically X and Y coordinates for each location. Then on each location, you measure gene expression measurement. So that's a count measurement, Y sub I here. And certainly, you also have a read depth, which is a normalizing constant, and an X, which are the covariates. So for the moment, you can ignore the N sub I and X sub I. It's just normalizing constant or something to control for. So because yi is a count, it's natural to use a Poisson or over-dispersed Poisson model to model that. So we'll assume that y sub I follows a Poisson distribution with a red parameter, basically a product between n sub I and lambda sub I. N sub I is just a normalizing constant, which is a read depth there, and lambda I is the unknown red parameter. So that's sort of the key parameter for inference in the Poisson model. And for the lambda I, because it's a positive number, so we usually model it at the log scale, and we'll model it as a combination of 4 different terms. So we have our usual intercept, usual covariates, and then the latter 2 terms, those are random effects terms. Those are the key for the modeling. So the B sub I and epsilon sub I are the key part for modeling the spatial versus non-spatial process. For the epsilon sub I, here we follow standard assumption. We assume it follows IID from normal distribution. So the epsilon I over there is trying to model the over-dispersion that you usually see from omics data sets. So you can think of this as over-dispersed Poisson model, very similar to a negative binomial model with the epsilon I over there. Now the B sub I there is unique. That one is trying to capture the spatial expression pattern. So to do that, we have B sub I for every I's location. So you can put all the n B sub I's together. So this is the n by 1 vector, the random effects on each of those measured locations. And then we model it following a multivariate normal distribution centered around mean of 0, and with the covariance captured by tau times k. So the tau is scalar, while k is an n by n kernel function of the spatial locations. So the k is essentially capture the distance on how those spatial locations are close or further away from each other. So intuitively, if I and j's location are close to each other on the spatial on the tissue, then the k sub ij is going to be large. And if k sub ij is going to be large, that means that b sub I and b sub j are going to be similar to each other. So therefore, the B vector here is trying to capture the spatial similarity and the spatial expression pattern. So therefore, identifying genes showing spatial expression pattern, you can translate it into a statistical problem of testing whether the entire B vector, the n by 1 vector, equals to 0 or not. And testing that entire vector equal to 0 or not is also equivalent to testing whether scalar tau equal to 0 or not. The scalar is a variance component. Specifically, if tau is 0, then the variance is 0. So when the variance and the mean both 0, then all the b's are 0. So the b's are basically, you don't have any spatial pattern. And if tau is non-0, then you can capture the spatial expression pattern. So therefore, all we need to do is to test the tau. Now, here, the K matrix is very important because the K, the kernel matrix, basically determines what type of spatial pattern the model is most powerfully to capture. So therefore, in order to capture a potentially wide range of spatial pattern you may encounter in real data sets, we look at a range of spatial kernels. So specifically, we look at 5 different Gaussian kernels with different bandwidths and 5 different cosine kernels with different periodicity patterns. So therefore, the hope is that you can capture a wide range of possible spatial expression pattern on any biological data sets you may encounter. So therefore, we're going to look at one kernel at a time. For each kernel, K, we're going to test the null hypothesis. There's no spatial pattern, so therefore the null hypothesis is that the tau sub K is 0. And we can perform inference based on penalized causal likelihood using the followed by average information algorithm for mixed model for reasonably scalable computation. And we can obtain a score test statistic for each kernel. And we can extract the exact p-value based on a mixture of chi-square distribution so that you can get an accurate p-value for each kernel. And after that, because you have 10 kernels, you get 10 different p-values. You're going to combine all the 10 different p-values through this Cauchy combination rule. So the Cauchy combination rule works pretty well, even though all the p-values are highly correlated with each other. So that way, at the end of the day, we are going to get one p-value for every gene, and that p-value is very well calibrated under the null distribution. So that's the main idea. So, so you essentially, just overall, in terms of schematics, so you have your goal is to detect genes show this type of pattern. You use the generalized linear spatial model to do that. You input 10 different kernels. For each kernel, you compute a p-value, and finally, you combine all the p-values together into a single p-value as indicated evidence for each gene. So that's the overall procedure here. So we performed.
Moving, can I ask you?
Oh yeah, please. Yes.
So this data contains a lot of zeros, like the counts. So it's a zero inflated distributions, right?
That's a good point. There's a lot of zeros, but it turns out it's not a zero inflated distribution.
Oh, is it true?
It's not. Yeah. So we look at the. That's a very good point because I think in the field, all the people would think because you have say 99% of zero, it's ought to be zero inflated. Mm-hmm. We actually fit a negative binomial model, Poisson model, zero inflated, negative binomial Poisson. All those models across, we examined, I think, like 15 different spatial transcriptomics technology. Mm-hmm. For majority of them, turns out the negative binomial is a good fit. Or sometimes Poisson is a good fit. There's no need to introduce extra zero inflated components.
Okay, so so what do? You add, so if you have the zero data, you add something, small number or something there, or no? You don't.
You don't need to do that. Yeah, because it's over dispersed. The Poisson is automatically accounted for that.
Okay.
Yeah.
The question is, these genes are correlated, but you are saying that if you do independent tests, and if you then combine with your Cauchy, I don't know what it is, and then it takes care of this correlation. Rather than doing a multivariate test.
Yeah, so that's another good point. So the combination is actually looking at one gene. You perform 10 different tests for each gene, and because the kernels are somewhat correlated to each other, so their p-values somehow correlate to each other. And the Cauchy combination, the loop basically is that you change every p-value back to a Cauchy statistics. Then you do a summation of the Cauchy, I think, and then convert the summation back to a p-value through a standard Cauchy distribution. So that way, even though the p-values are somewhat correlated, it turns out that this adjustment is reasonably robust in terms of correlation. But I think we are raising another important point is that all the genes, some genes are correlated with each other due to genetic gene co-expression or gene networks, right? And for that, we did not account for that. We simply treat every gene independently analyzed, so you get a one p-value at a time.
No gene space interaction there.
There is no gene space interaction exactly in this model, but we did look into this in the supplementary figures. We try to borrow information across multiple genes that share similar patterns. So that way, it's essentially kind of hierarchical framework we build up in the supplementary tests, where you can think that we assume that some genes, they share certain spatial expression pattern, and we try to borrow them to get a better p-value. And turns out it's a bit more challenging to do. You can get a p-value. The p-value is just not as calibrated as if you treated them independently.
How bad it is, like you're having a hierarchical which is perfect spatial model. But if you take just yi, when zero doesn't have much effect and make a transformation, all biologists do that, right? Make it log yi. Then log lambda into log yi. Add a small constant to the zeros, and then put a Gaussian processing for that. Is that gives completely wrong results, or
good point. So we did that. So we have a Gaussian version of the Spark actually, just treating normalize the data as if it's a Gaussian distribution and you fit everything. It turns out that because of the count is very low, you have a lot of zeros. So if you don't use over dispersed Poisson, your p-values are not calibrated. You're going to see a lot of false signals basically. And so that's one issue. The other issue is that when you have a large data set, when you fit this type of Gaussians, I think you also have a deflation. There will be a figure later on, not in this section, but in the second section of the probably point to that. Yeah. 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 And please feel free to interrupt. I would love to answer questions.
So for the zero inflation part, I know you said that it seems empirically you don't need a zero inflation.
Right. Yeah.
Binomial.
Yeah.
I mean, intuitively or biologically, why is it because their lambda is always very small, so you always get excessive amount of
good point. So the way we think of it is that first, if you have a lot of zero, if the lambda is very low, if you just simulate from a Poisson distribution, you ought to get a lot of zeros. If you simulate from a negative binomial distribution, you ought to get zeros. And the reason that negative binomials seem to be reasonably good is because that over dispersion is trying to capture the heterogeneity across different cell types. And across different cell types, the zero inflation is a very, very strong assumption saying that underneath of that, some of the lambdas are exactly zero, some lambdas are small, while the negative binomial is more like general, every log scale lambda follows sort of normal-ish, not exactly normal, but a gamma distribution, I think, something like that. So it's much more sensible assumption, turns out, for many biological settings. That's the main reason. But that's one technology that zero inflation does seem to work very well. So that's, if I remember correctly, that's a Murphy technology. So somehow they do have a zero inflation issue, and you do need a zero inflated negative binomial in order to properly model that. We do not understand why that's the case yet. Yeah.
In that, I think the fish technique, you have like almost bimodal. You have zero and mode and zero, then you have a mode that's at 10. If you have like two modes, then negative binomial or over dispersed,
right?
So I'm not going to fit there.
Right. Yeah. So Murphy.
Try to plot and doing a mixture of all the computational good things, right?
Yeah.
If you have this unimodal, it's probably fine. You just have a small lambda for over dispersed one.
Right.
Account for that. This bimodal or even trimodal.
Yeah, if you see the bimodal, and also you want to be a little bit more careful when you look at these patterns. You want to visualize raw data versus normalized data, because normalized data, even if you generate from Poisson distribution, once you normalize it, you will artificially introduce bimodal patterns too. Yeah. Yes.
I wonder if you can see the spatial pattern by looking at it, like looking at the
Yeah.
data.
That's how we usually validate it by looking at the data. We'll see.
Really, they have a spatial correlation.
Yeah, I'll show you some of the examples. Yeah.
Simple,
Maureen side test. We tried that too. Yeah.
Compare with those simple.
Yeah, we tried that too. We put this results into a discussion paragraph. The simple test, the p-value somehow they're not calibrated. So I think we are. I'll show you quickly later on the p-value plot, like here. It's extremely stringent. I think we're looking at to see whether it follows uniform 0-1 distribution. So those simple tests doesn't seem to work. We do not understand why. Yeah. So it seems to be So I'll show you quickly there.
You use the negative binomial distribution is over dispersion, right?
Yeah, it is over dispersed.
Multinomial distribution.
Oh, we haven't tried that because multinomial, I guess you are thinking of modeling multiple genes jointly. And because for each gene, because across all the genes, I guess it's natural to think all the genes will follow a multinomial distribution. And then marginally for each gene, that sort of follows the binomial distribution, right? But modeling across all the genes, it's challenging computationally mostly. Yeah.
And one more question, since we're talking about this. So you try 10 different kernels, and you're saying, oh, with 10 different patterns, it will combine the p-values.
Yes.
And so they might be correlated, fine. But in reality, you will see maybe it's a ring or maybe it's more Gaussian.
Yeah.
So how sparse are the p-values now? Are they really that correlated?
So the p-value, so we didn't look at the p-value correlation, I guess. We only look at a histogram of p-value because for each gene you only get one p-value. I don't know how to check the p-value correlation.
For each gene, you have 10 p-values of the current 10 kernels.
I see.
How
they correlate, I see. Yeah, we haven't looked into that. I suspect some of them are going to be correlated because like some Gaussian kernel, when you have similar bandwidth, one is slightly small and slightly large, they're probably similar to each other.
Yeah, because I was thinking maybe mean P will give you the highest power because
that's possible.
One is really extreme, the others are almost all ones.
Yeah, that's possible, but we didn't choose that for two reasons. One reason is because the p-value calibration, as I'll show you on this left figure, the p-value calibration is extremely hard to achieve. The second is that for the Cauchy rule, it's also somewhat captures the minimum p-value because if one of the 10 Cauchy statistics is very strong, you automatically the summation of them is turned into a p-value that's somewhat similar to that p-value essentially. Yeah. All right. Okay, so let's look at this real data example. I'll skip all the simulations. So in the real data example, this is spatial transcriptomics sequencing technique. We did a very minimum filtering, filter out genes that are expressed in less than 10% of the spots, select spots with at least 10 total reads. In total, we analyzed 5,000 genes measured on 250 spots, right? So we look at two things here. One is the type 1 error control, the other is power. So type 1 error control is on the left-hand side, power is showing on the right-hand side. So for the type 1 error control, what we did was to do a permutation, because once you're on the Y's, once you permute the Y's, you would expect all the p-values are non-significant. So the p-values under the null will follow a uniform 0 to 1 distribution. And in order to, because we care about small p-values, so we did this minus log 10 transformation so that the larger number you have, the smaller p-value you have. So we're plotting the observed one versus the expected one. So if everything works well, you would expect the method sits on this diagonal line. So you can see that our method, the spark follows on this diagonal line. Now the spatial D is somewhat to the Gaussian version of our method. It's doing some Gaussian transformation. So you can see that initially it's very conservative below the diagonal line. So it overshoots, meaning that it identifies a lot of signal in the permuted data set, which is not ideal. And then for the Transcriptomics, most of its Transcriptomics versions works fine because it's using a permutation-based test. So it's extremely slow, but in terms of type 1 error calibration, it's okay and too conservative. And so because of this huge inflation of spatial D, it makes harder to compare the power. So we compare power based on the same force discovery rate, which is estimated by the empirical distribution of the null. So you can see that our method is indeed much more powerful than the other two methods. It's 2 to 3 times more powerful than spatial D and about 10 times more powerful than Transcriptomics. And so whenever you see this type of power gain, we initially we don't believe it because in genomics, usually our experience that if you get 20% of power, that's great. That's going to be a good paper already. 3 times, it's probably did something wrong here. Usually, that's our first thought. So we did a lot of careful analysis, including, for example, we checked the Transcriptomics, what's going on with the low power. We use their own simulations. We found that in their own simulations, we are able to recapitulate their also power simulations, but it turns out their powers was they can achieve 60% power, but all the other methods can achieve 100% power. So they basically have to create a very strong patterns in order to achieve a good power. It's just not powerful enough. So we did a lot of more rigorous analysis. So the first thing we did was to look at some of those known genes. So there are 10 known genes in this data set. And among the 10, turns out that Spark can identify all the 10, while the other 2 methods combined can only detect 7. So this is a tissue, what tissue looks like. So this is a cancer tissue. This is actually an E staining. The bottom part, where the dark area is essentially a cancer area, while the top, the white type of area is a normal tissue. And so you can see that for those genes, each of those genes, those are the cancer associated non-cancer genes. So the p-values are pretty good from Spark, and the patterns are quite obvious, at least for majority of them are enriched in the cancer area. So the second analysis validation we did is to look at the first the overlap. So we found that we focus on our method versus spatial D because spatial D actually identified a decent set of number of genes. So majority they are overlapped, but we did identify 200 some genes that are unique, and spatial D identifies 30 genes that are unique. So therefore, we divided genes into 3 sets. This identified by us, commonly identified both us and the spatial D, and uniquely identified by spatial D. And we simply ask, among those genes, say 75 genes, how many of them have been existing in other existing cancer database? like we look at two different cancer database, cancer mind database, which is small ones, as is relatively large one, hormonal database. So in the overlap set, 75, you can see that 54% of those genes exist in this cancer database. And then if you look at our unique one, that's a 43%, it's lower, but it's still reasonable. And then if you look at the spatial de-identifying, it's even lower. So this also suggests that the genes, even though these are uniquely identified by our method, these are also seem to make sense. And here shows some of those example genes that are uniquely identified by our method. So the first one is the ERBB2. So this is a well-known transcription factor for breast cancer. So ours was able to identify that. So it's very reassuring. The second is that we were able to identify the top gene is HLA-B and then the CD44. Both these two genes are immune-related genes. Those also make sense because it means that the immune cells start to infiltrate to the cancer regions, start to fight off the cancers. So it's nice to identify this related immune system-related gene. And then finally, we also identify EF1A1, which is the elongation, cell elongation factor, also MMP14, which is the extracellular matrix. And both are known biological processes associated with cancer, especially with cancer migration, because you need extracellular proteins to sort of dissolve and in order to make the cancer migrate, and those elongations, those sort of process captures the migration of those cancers. So it's very reassuring to capture all those detailed biological examples. And we also did a GSEA gene set enrichment analysis on the genes identified by our method. You can see the top signals are all make biological sense: extracellular structure organization, extracellular matrix, collagen, response to cytokine, regulation of immune responses. So they all seem to make sense. So, therefore, you can see that Spark is indeed quite powerful and help you understand the biology of those spatial transcriptomics tissues. So that's Spark. So when we developed Spark, that was published in 2 years ago, I think. But in the past 2 years, there was a huge progress in terms of spatial transcriptomics technology. Specifically, the spatial transcriptomics technologies becomes really, really large. So there's a huge increase in terms of the number of spatial locations you measured. So those recent technologies like the slide-seq, HDST, are of very high spatial resolution, so we can measure tens of thousands or to hundreds of thousands of spatial locations. So this is different from the setting when we develop Spark, where we only have typically see hundreds of spatial locations being measured, or maybe occasionally a few thousand spatial locations. Now, a couple of years ago, it's extending to tens of thousands, hundreds of thousands spatial locations, and nowadays it's not rare to see tissue sections with millions of spatial locations. So there are, with the increase in sample size, because the sequencing depth is fixed, because the budget is fixed. So your sequencing depth is fixed. So as a result, this data are extremely sparse with a prevalence of low counts and a lot of zeros. So that creates important computational and statistical challenges. Specifically in terms of sample size, which is the number of spatial locations here. And it turns out that all the methods we have been developed, the Spark, all the previous methods like spatial D, Transcriptomics, they all have cubic computational complexity and also cubic memory requirement, which is problematic because now it can take days to even months to analyze one data set, and it will cost dozens of thousands of GB memory requirements. So people just with a desktop, you just won't do it, won't be able to do it. Even like us, we have access to the one of those largest computing facilities in the US, we probably won't be able to do it either because of those huge memory constraints. So that's the issue with the increasing number of locations. The second issue is the low counts, the prevalence of zeros. So we try to model the counts directly with positive and negative binomial, which is fine with small sample size. But it turns out that once you have a large data and if you have a lot of sparse, a lot of zeros, it will fail this algorithm. The standard negative binomial distribution, if you just look at R package, standard negative binomial distribution in R package, if you simulate data, we say over 90% of zeros for the gene from the negative binomial model, if you try to fit it, it will fail to converge in majority of those cases. And in fact, in many cases, I think it's over 90% of genes it will fail. Sometimes it's over 98% of it will fail in over 98% of genes. So it's a lot of problem in fitting this negative binomial type of or over dispersed type of Poisson model. And then alternatively, if we try to normalize the data using, say, Gaussian to model that, like the Gaussian version of Spark, it will result in power loss and also a failure of type 1 error control, especially at a small p-value. So you tend to identify a lot of false signals if you normalize data and using Gaussian to model that. So therefore, motivated by all those issues, we decided to develop another version of Spark. We call it Spark X or Spark Expedited. So this is a completely different modeling framework. So it's based on a general class of covariance tests that's for non-parametric spatial expression analysis. So the main idea is this. So we're going to construct two kernel matrices. One is the gene expression kernel matrix E, which is n by n. The other is distance kernel matrix sigma, which is also n by n. So the intuition is that if I and j's location are close to each other, then the distance kernel, certainly the sigma ij, is going to be large. And if this gene is indeed a spatially expressed gene, then you would also expect the Eij to be large. So therefore, you would expect the E matrix and the sigma matrix, each element of in the E matrix, the sigma matrix, they are going to be correlated with each other. So therefore, you can just do the product between these two element-wise, which is basically a trace term of E times sigma, and that statistics will be large. So, therefore, we just need to compute these two kernel matrices, get their product, treat it as a statistics, and extract the p-value. But certainly in the process, we need to perform a range of nonlinear transformations on the coordinates in order to capture a wide range of possible spatial patterns. So we also performed about 10 different nonlinear transformations over there. And then we combine, for each transformation, we compute a p-value, and then we combine the p-value across this 10-ish transformations through, again, the Cauchy combination rule. And in the process, which is very critical, is that we develop a lot of algebra innovations or manipulations to ensure the algorithm actually scalable. And this is really challenging to do. But at the end of the day, we was able to do that, and we was able to reduce both the computational complexity and the memory requirement from cubic to linear. So that's a huge difference. That's an n square savings difference. So you can quickly see how that goes here. So we're plotting the time, computation time versus the number of spatial locations, and also memory requirement versus the number of spatial locations for 4 different methods. The newer versions, the red one, the Spark X, the other is Gaussian version of Spark, the Spark and the spatial DE. So Spark thought is over here. It's using a Poisson, over dispersed Poisson, so naturally it's slow to fit. So for a few thousand locations, it's fine. For 10x reason, it's completely fine. But once you have 10,000, 20,000, it becomes very slow because it's cubic increasing. Even the Gaussian and the version of Spark or the spatial DE are very slow. So as you can see the clear cubic increasing pattern. So they can probably handle 20,000, 30,000 spatial locations, but not at 50,000. While for our method, you can see it's linear, basically. It takes less than a second potentially in those example cases. And it also uses a very limited memory, unlike other different type of methods. So therefore, Spark X is extremely scalable. So we can look at some example data sets. One is Marcel-Baron data, the slide-seq data, which measured 11,000 genes on 25 spatial locations. So in this data, you can see that in terms of computational cost, Spark X is very low. RAM cost is also extremely very low. And you can look at type 1 error control. So in this, we again permuted the data. So you can see the Gaussian version of Spark, which when you model the normalized data, it's really bad. You see a huge inflation of p-values. You identify a lot of false signals in the permuted data, while Spark X is fine. And certainly, if you are able to run Spark, it will be also fine. It's just not that scalable to handle 25,000 spatial locations. And in terms of power, our method still works much better than this Gaussian approximation version. And you can validate with overlap with the existing databases also. So there are some gene examples I'll quickly skip. So this type of analysis, one unique feature of Spark X is that because it's so scalable, it allows you to quickly run additional analysis that can help you answer additional important scientific questions. So one important scientific questions we saw at that time was to figure out what are exactly those spatially expressed genes. And because intuitively, right, if a gene is a cell type marker gene, because the cell types are always show spatial expression pattern on the tissue, then you would expect the cell type marker genes to show spatial expression pattern. And so we're wondering, among all those genes we can detect, how many of them are actually, say, cell type marker genes that show spatial expression pattern simply due to the spatial distribution of the cell type, and how many genes are not. So to do that, we simply include those cell types as a covariates into the model to adjust for them, because intuitively, after you adjust for cell types, then you are only going to detect genes show spatial expression pattern not due to the spatial distribution of the cell types. So that's what we did. So we basically did two analyses. One is original analysis, that's a big circle. We identified 520 different genes that show spatial expression pattern. Then you control for the cell types. And then you find that half of genes are gone, half of genes, 260 genes are no longer significant. So those are potentially the cell type marker genes. They're no longer significant because you control for the cell types into the model. But there are still about half of genes left. So we decided to look at some of those half of genes to see what's going on. Why do they still show spatial expression pattern even you adjust for cell types? So here are some examples. So on the top, it shows, so the first row shows the cell type distribution. So we're showing on top the granular cell type distribution, and the bottom the Purkinje cell distribution. So it's all colored blue, this particular cell type. And if you look at the right column first, so the CAD-M3 and CALB1, those two genes are essentially the cell type marker gene. The top one is the granular cell type marker, the bottom one is the Purkinje cell type marker. And you can see that the p-values, initial p-value is significant, but after controlling for cell type, it's no longer significant. Bottom is also initial p-value significant, after controlling for cell type, it's no longer significant. So that's as expected. Now, the middle two part is that they are most significant before the initial analysis and also after for cell type control. It turns out that these genes are the genes where show subcellular, shows the spatial pattern within a particular cell type. So specifically in the top, the PT, PRT, it shows expression in the first half of the granular cells spatially, but not in the latter half, while the LDOC, it shows heavy expression in the latter half of the Purkinje cell, but not the first half. So that's indicating that cell balance is really spatially compartmentalized in order to carry out different biological function. And those signatures are the genes that characterize the transcriptomic basis for this cell balance compartmentalization. So finally, I'll quickly go through this factory Bobby example, because this example is the only one that that our method is the one that only our method works. So it measures on 177 spatial locations. That's considered as relatively large a couple of years ago, but nowadays it's small at a scale actually. So you can see that in our analysis, we can identify a lot of those example genes, and the top pathways, they all make biological sense. So that's basically the SE analysis on Spark X. So finally, I'm going to, through the sort of switch gear talk, hopefully use another 10 minutes-ish to talk about the cell type deconvolution we recently worked on. So the question, the scientific question is that, so a lot of people nowadays, when you perform spatial transcriptomics, the easiest way is essentially to use 10x vision, because this is a commercially available platform. You can directly use it. You can measure 5,000 spatial locations, which gives you pretty reasonable spatial resolution. But the issue is that the 5,000 spatial locations, they are not at the single cell level. So they're essentially measuring a spot that contains about 2 to 20 different single cells. And so therefore, in a way, it's not of high spatial resolution. You do not get down the cell type levels. So essentially, in terms of image, the truth is probably like this. You have a lot of single cells measured in this dot, but you are essentially get a blurry image. You are only getting average of that. So ideally, once you get this type of data, if you can tell the biologist that, well, in this measured spot, there are actually 5 different cell types are there, and you can have different compositions for them. You have 25% for cell type A, 50% for cell type B. If you can get biologists this type of information, I think this will be extremely valuable. So they can ask additional, more important scientific questions. So therefore, at that time, we decided to develop a method to do this type of cell type deconvolution, extract the cell type composition information on each and every measured spatial location. So to do that, we try to integrate 2 different data types. One is spatial transcriptomics, which is your current data you have. The other is spatial, is a single cell RNA sequencing, which are commonly available in many different public domains. Because the single cell RNA sequencing gives you unique information on the cell type specific information, and this you can incorporate into the model in order to perform spatial deconvolution. So there are multiple different deconvolution methods were developed. So at the time we developed methods, there was only one. So that's only for bulk RNA sequencing. And during the process, I think there were 5 methods developed for spatial transcriptomics right before we submitted our paper. But we did notice that there are multiple limitations of those methods, including those methods that ignore the spatial correlation among cell type composition in neighboring locations, because intuitively, if 2 neighboring locations are close to each other, you might expect they share similar cell type compositions, and we can actually borrow information across neighboring locations to do a better job to do a deconvolution. Second is that all those methods that rely on a reference panel. So that's our initial thought. We try to use a single cell reference. But it turns out that you don't even need that single cell reference. If you only have your spatial transcriptomics data, for whatever reason, if you cannot find a match to the single cell, it's still fine. You can still use our method to do deconvolution. And finally, it focused on those methods all focus on deconvolution. And turns out that our method can do much more than deconvolution. So this is a model. So the model looks like this. So you have gene expression that's in the X. It's essentially a factor model. And the B is the single cell reference, the reference matrix. And while the V is the cell type composition. So we express X as a product B times V plus some certain of residual error term. So the standard factor model is B known when you have a single cell reference. Certainly, if you don't have a single cell reference, you can also try to infer B directly, as we also did over here. So the unique part of this model is that for the V, we try to incorporate neighborhood similarity information so that you can do a better job to estimate cell type composition. So specifically, we specify a conditional autoregressive model assumption on each column of V so that you sort of encourage the spatial similarity in terms of cell type composition. And so this way, you can do a better job at inference first, but secondly, more importantly, by borrowing the neighborhood information, you can actually do imputation on both gene expression, gene cell type information, but also on gene expression. So, finally, we develop a constraint optimization algorithm in the maximum likelihood framework to estimate the cell type compositions. So here basically shows the overview. So we have a single cell reference, we have our spatial transcriptomics. You try to combine them together. But as I mentioned, sometimes if you don't have a single cell RNA-seq reference, it's fine. You can just use deconvolution with the factor model to learn both B and V on the spatial transcriptomics. And that's a model. The key part is to use a CAR framework as sort of as a pry on the V matrix. So therefore, we call our method CAR, the CAR-based deconvolution. And you can estimate. So the key product of this method is that you can estimate cell type composition on each measured location. So now for each measured location, you can express it as a pie chart. So each different color represents a different cell. The size of each slice represents a composition or proportion of each cell type on this particular location. And because of this this conditional autoregressive modeling, the borrowing information about neighborhood, that allows you to do imputation of both cell type composition and gene expression on unmeasured locations. So if you only measure 5,000 locations, then you can try to impute what's going on in the middle among them. So therefore, this allows you to construct a very high resolution spatial map. So let's look at a real data example. So this is a cancer data, pancreatic cancer data. So this is spatial transcriptomics with 25,000 genes and 400-ish locations. We have a single cell RNA sequencing reference. And here shows the results. So it shows 6 different methods on the left and the card on the right. So the pie chart on card, you can see that it sort of captures the general structure of this tissue. And more importantly, it's able to capture 2 distinct cancer subregions in this particular tissue that's not captured by other methods. So specifically, on the top one, you can see that this is sub-cancer region that's dominated by the green cells. The bottom one is dominated by the pink cells. And turns out that the pink and green cells are 2 different stages of cancer that they do express in 2 different regions. So specifically, that's a clone A cells that tend to express in the top half of the cancer region, and then clone B mostly enriched in the bottom half. So therefore, through deconvolution, now you are able to capture those different subdomains of those cancer regions. And we can certainly compare with sort of ground truth the single cell RNA cell type composition to see which method works best. So you can see that our method achieves the highest correlation comparing with the ground truth, the average cell type composition compared with the other methods. And it also can do a spatial refinement. So instead of blurry image that you measure with 10x vision, now you can get a much refined image. So specifically on top, it shows the different cell type compositions. Once you refine it, you can see that it's a clearly spatially refined map of the cell type composition as well as gene expression. And you can also do it without any reference. So this is a reference-free. Certainly reference-free because you don't have a single cell reference, you do lose a little bit more information. So in this case, although the general structure looks very similar, you do lost the subdomains in the cancer region. And finally, the method is very fast. So it's in terms of computation time, it's the best method among all the competitors, even comparable with music, which is a bulk RNA sequencing based method, which is known to be bulk is easy to run. And in terms of RAM, it also uses quite small compared with many different competing methods. And importantly, just after our paper published, there's a comprehensive benchmarking study carried out by a different research group. And in the benchmarking study, we compared 20 different deconvolution methods. So this bar, the gray bar, basically shows which method works the best overall in terms of 20, 30 different criteria. And you can see that the card is indeed works the best one among all those ones. This is verified by an independent research group, which we were really happy to see. So finally, as a summary, so basically, I talk about 2 different topics. One is the SE analysis, the other is cell type deconvolution. For SE analysis, we introduce Spark and Spark X. They are effective methods for detecting genes that show spatial expression pattern. And then for deconvolution, we introduce CARD, which enables spatially informed cell type deconvolution for spatial transcriptomics, either with a single cell reference data or without single cell reference data. And all the details are available on this 3 paper, and the software available on our lab website. And finally, I would like to thank it's all really done by the talented postdoc Sichuan and the 2 talented graduate students, Jiaqiang and Yin. Both are over here. And that's all. And certainly we are funded by NIH, and we have postdoc positions. See if you are interested in applying. All right, so that's all. Thank you very much.
Questions? We have already had a very good though, but any last minute question?
A quick question on the spatial deconvolution.
Yeah.
So you have a non-net effect rate.
Yes.
The B matrix is known as treated as known, or is it?
Yeah. So in the single cell reference based approach, the B is treated as known. You extract it from a single cell, but in the CARD-free, the free version, the reference-free version is treated as unknown. You infer the B.
If it is unknown, then you need to identify it.
So we do have different constraints on the B and the V. So specifically for the B, I think even though it's unknown, we still tell them what are the cell type markers. So you still need to know the cell type markers in order to run the B. So that's one sort of constraint. The other constraint on the V is constraint to be between 0 to 1, sort of to make up since it's a proportion. Yeah.
Any other ones?
Just from a
technology perspective, or more like data harvesting. So in order for the spatial, I guess, variability and transcription to matter, like you need to have a biopsy sample that sort of covers a large spatial pattern as well. Like you have to work with doctors, or is there a change that in the biopsy pattern so that you're actually selecting both healthy tissue, cancer tissue, different types of cancer tissues? Because if it's all uniform, kind of target with biopsy a particular cell type, then maybe the spatial variability is a priori limited.
Right. Yeah. I think it's very important to see capture the tissue according to a scientific question. So if you are interested in say cancer, I think nowadays we are interested in extending those methods to settings where you are mentioning where you have multiple control samples, where you have multiple cancer samples, because that way you can actually model those heterogeneity through across samples. But the main issue is still it's very expensive technology. So multiple samples data sets are still relatively rare nowadays. But I think that will change very soon in the next couple of years.
So due to the time limitation, let's start our